Hi everyone, welcome to the Tokyo College UT7 interview series. I'm Chunyan Li, a postdoc at Tokyo College, and today we're very glad to have this opportunity to interview URANO Yasuteru sensei about his research in chemical biology. My research focuses on how we use digital technologies and social determinants of health to change or to promote the health and specifically sexual health and HIV prevention among LGBTQ people living in Japan.
Okay.
And we're very glad to have this chance to talk with you about more about your research and the UT7 program. So first of all, would you please share with us a little bit about your research and your work in plain language?
Okay, so it's so difficult to say in plain language because that I'm a chemist actually. So I'm a professor in this medical school and also a professor in the pharmaceutical science department, but I'm the chemist. So to synthesize the new types of the functional molecules. So one of the functional molecule is to lighten up the cancer cells among the normal cells. So the surgeons want to know where to resect and where to be remained. So but during the surgery or during the endoscopic surgery, so it's not so easy to find the cancer cells very selectively. That's why we developed a kind of the functional probes. We call it the fluorescence probes. So the fluorescence probe itself is not fluorescent actually, but by reacting with the cancer cells, it turns to the highly fluorescent product.
I see.
So that's why we can stain the cancer cells very selectively by spraying these probes to the cancer suspect regions. So this is one of the way to help the surgery or the endoscopic diagnosis with chemistry.
I see.
So this is the one typical our achievement actually.
Yeah,
so
that is very interesting. I read your blog when before I came here. So it talked a lot about like personalization of medical treatment or precision medicine.
Yes.
So would you mind telling us like what are the biggest challenges in this field?
Our main goal is to overcome the cancers, actually. So for overcome the cancers, it's quite important to know exactly about this patient's cancer cells. So this is also called personalized medicine, actually. So as you know, the cancer is a heterogeneous disease. So we need to know exactly about the nature of the cancer cells of this patient.
Yes.
So that's why we have already developed plenty of the fluorescence probes, maybe more than 1,000 kinds.
Wow.
Yeah. So that each compound, each fluorescence probes detect the different enzymatic activity.
I see.
So by spraying the probe to the cancer regions of the one patient, if the cancer cells is brightened up, these cancer cells have this specific enzymatic activity. So we firstly know that this patient has the very heterogeneous, of course, cancers, but the measure of the cancer cells have these types of the enzymatic activity. So this is a kind of the personal medicine. And by utilizing this enzymatic activity, we will succeed to develop a kind of products, so which kill the cancer cells very selectively. So this product itself is non-toxic itself. So, but if this molecule encounters to the enzymatic activity, which is very specific to the cancer cells, it turns to the very toxic compounds. That's why this product can kill the cancer cells very selectively. So, in this meaning, first we know by spraying the probe, maybe the 10 kinds of the probes is enough, so to know what types of enzymatic activity is very specifically upregulated for this patient's cancer cells. And second, to apply the product to the sense and actually to have the kill capability of the cancer cells, so one by one.
I see. So you mentioned you developed, or the field has developed more than 1,000 types of the fluorescent probes.
Yeah.
So each is designed for a specific type of cancer, or?
Yeah, then actually our probe is capable of detecting the enzymatic activity itself.
I see.
So there are, for example, the proteases, as you know. So the protease can cleave and hydrolyze the amino acid sequences. So our probe detect this enzymatic activity one by one. So first, we must know what types of the enzyme activity is actually upregulated in the cancer cells compared to the normal cells. But it's not so easy because so there are, of course, a bunch of the cancer cells, actually the cultured cancer cells. But the cultured cancer cell is originated from, of course, the human patients. But as you might know, the usual cancer cell cannot live long if we incubate it say, in the media. But the very small percentage of the cancer cells can live long, and then we call it the cultured cancer cells.
Yes.
So this cultured cancer cell is, of course, one of the nature of these cancers, but maybe the majorities are not. So that we need to know the nature of the majority of the cancer cells that this patient has. That's why we spray the probe itself. So the probe can detect the enzyme activity. So we first know that this patient has this specific cancer, the enzyme activity in the cancer cells. So this is very completely different strategy to know the nature of the cancer cells of the patients.
That sounds very interesting. As you mentioned, maybe some of your tests or experiments are done in the culturated cells. So how is the application in the clinical practice now?
So now that we are now doing we already finished the phase 1 study, and now we are going to the phase 2 study by collaborating with the endoscopist in the University Hospital Tokyo.
Cool.
So we already finished the toxicology test, preclinical test for the specific the fluorescence probe, which can detect the esophageal cancers. So now we are doing at this stage for the phase 2 study. We have a lot of the other achievements for finding out the appropriate fluorescence probes for each cancers. For example, the glioblastoma or the stomach cancers and the colon cancer and so on. So each cancer cells have a little bit different the specific enzyme activity. That's why we need to do the more and more preclinical tests and then apply for the human patient as a phase 1 and phase 2 study.
Of course. Well, that sounds very exciting. And you sound very passionate about your own work. But I believe as a scientist, myself as a researcher as well, so there are some unhappy moments or obstacles as well. Would you mind sharing us?
Yeah, actually, I don't know it is exactly for the unhappy moment, but we need to have the permission by the government for commercializations. So this kinds of the how can I say, but the probes to lighten up the cancer cells is the very first type of the functional molecules. So that there are no approved probes in the world for that. So now we are now applying for the Japanese FDA. So, but they don't have any experience to tackle with this kind of thing. So that they don't know what types of the achievement, what types of the proofs are needed for the commercializations. So that's why that we are now arguing each other. So that it's very tough way, and actually then it took so long time. So. But we hope that we will overcome these types of situations in the near future, and then we can commercialize it. And then after the commercialization, of course, all of the surgeons and the endoscopists will utilize these types of probes. So there are for the surgical procedure, of course, as you might know, the 100% resection proves the complete curement of the cancers. But if 1% is remained in the body, so it's what's called a recurrence. So the recurrence is a very heavy problem for the patients. So the surgeons and the endoscopists can find out if the less than 1% of the cancer cells by spraying our probes. So that must be the very hallmark for the patients, actually.
Yeah. For ethics-wise, we definitely need to put the safety of the patients first before in front of any kind of research or experiment. But in terms of the negotiation between the regulation authority and the researchers, how is the university playing a role in terms of supporting our own researchers or professors?
So it's not so easy question actually to answer. So, but I believe that, and I hope in the future that the university must have the function for helping the peoples in the world, and of course in Japan and in the world, to keep their good health for a long time. So these types of the achievements, of course, we are the researchers, so that we want to write the manuscript and publish it. And so, but that after that, we need to commercialize it and make more people happy with our probes and with our strategies and the technologies. So in this meaning that university can help these types of commercializations and socializations of the functions, functional probes for them. So in US, so there are so many platforms built in the university to help the researchers to commercialize and to make it socialize their new types of the technology in the world and in the US. So maybe our university can do these types of the function, of course. So that maybe the Fuji Sensei will have these kinds of the motivation. So in the near future, our university must have the more, how can I say, the commercialization for these types of course, the drugs and the technologies and for the people in Japan.
It's like to disseminate the research findings from the university to the broader society.
Yeah, of course, dissemination is important, but the more important is to let the people can use directly. So commercialization, so dissemination is a kind of information-based the disseminations. So of course they have the strong will and they have a strong interest about it. So, but to make the people feel that our university can help the level of the life. So in Japan and in the US, in the world. So yeah, we need to commercialize the probe or the drug itself.
That's
and
true.
to make them the usable for the peoples. So maybe the information is the first important way, but after that we need to, how can I say, but
make it more sustainable and accessible.
Right.
Yeah, that's very interesting. That's very important. So I think, is that something UT7 is going to do?
Yeah, actually the UT7 has roughly have the two major project goals. One is to know what is a life, actually. So this is very research-based, actually.
I see.
So the other part of the project, and actually I'm involved into the latter part, so this makes the people more happy. So make the life more happy. So in this meaning, of course, as you might know, that there are so many expensive drugs. So like 50 million yen per dose or something like that. So of course, this is the very state-of-the-art the technology to cure the patients. But of course, the expensiveness is not good information for the majority of the people. So we need to develop the more cheap but more highly functional drugs for that. So in this meaning, that I believe as a chemist, the chemistry can do these types of the help and the achievements for that.
Yeah, chemistry can help we develop more cost-effective tools.
Right.
Like we can maintain the high quality, but as we lower the cost to make it more accessible to people.
Actually, I mentioned that they usually intake the aspirin when you have the headache. So if the aspirin is the 50 million yens, you don't have that. But we can buy it maybe the 1,000 yen or something like that. So that's why we can take and we can cure the headache by very easy way. So in the future, to overcome the cancers, we need these kinds of the very cheap, but not so expensive, of course, and the highly functional, which means that the sensitive and selective for the cancer cells. So that's our goal, and the UT7 will do these kinds of the basic research, of course, but heading for the helping the people and the keeping the high level of the health situation especially in Japan.
I see. So I feel like that the second goal, the part you're involving with, like how to make people more happy, not only in Japan but all over the world.
In the world.
Yeah, that's not only about basic science research, but also should involve more researchers, more experts from humanities and social science.
The social science is very important.
But so far in the portfolio of the UT7 program, it seems there is no professors from or researchers from humanities or social science. How do you think about that?
So of course we are now arguing about this point, and then we already had a lot of the discussions and meetings with the social scientists here. So, but they don't have actually at this moment, so that they don't have so much interest to involve in UT7 because we cannot show them that this drug is, of course, the first target, and this drug, the second target, something like that, more concrete way. So if they know, these types of drugs are very effective for keeping the high level of the health in Japan and the world and for the people, so we hope that they will join the UT7. And actually, some of the young researchers in the social sciences have a strong interest about it. So that maybe we must, of course, disseminate the information for the social scientists with these types of achievements in the science.
So maybe we need to create a dialogue, something like this interview to communicate what you're doing in the UT7 program to a broader audience at the university, so maybe they will be interested and then attracted to join this program. That would be nice.
Yeah, so in this meaning, actually, I'm sorry that I don't know much about the Tokyo College system. So if this kind of the system can help us to disseminate these types of information and involve the social scientists and the other scientists, so we want to increase the number of the professors in the UT7.
So Tokyo College, actually, I think we're a multidisciplinary team, and we have a lot of research experts from not only within Japan but also out of Japan to come together and just everyone working on their own research, but also we have the time and space to chat with each other and maybe get some inspiration from each other's communication. So I think, since we're all based in the university, and my own personal understanding of a university is it's the place we educate the next generation of the global leaders or the scientists. How do you think about the students' training at the university environment?
Not easy question to answer, but in my opinion, I don't think that we need to have a lot of the classes in the university for teaching the students. Of course, in high school and junior high school, we need to teach very basic science, chemistry and physics and mathematics and so on. So, but after entering into the university, they must find out their own interest. So among the vast of the science or of course including the social science. So in this meaning, in my opinion, so the main object of the class is to share the passions of our scientists with the young generations of the students. So if small percentage of the students have interest to share the passions of our scientists, as for example, for me to make more happy life for the people. So they must find out some way to tackle against the problem for that. And then in the Faculty of Pharmaceutical Sciences or in the Faculty of Medicine, so they must have some their own project in the very near future after that. So in my opinion, the classroom must have this kind of function to share the passions.
I see. So it's more passion-based, to inspire their own motivation to pursue the science.
Of course, we can teach the very basic chemistry.
Yeah, the skills and the knowledge.
But this is a very small part, actually. I believe.
I see.
So the majority is to feel the passions of our science motivation. Actually, some of our PhD student is a surgeon, actually.
Oh.
so that they have, of course, their own. So they usually they usually do their own surgery every day. But after the surgery, so that they usually come to my lab and then to do some experiments.
It's very busy.
Yeah, so busy. But they have the strong passions. They know that the insufficient tools, so on, for doing the very precise surgery. So that's why they have the strong passion to develop the new tools to help them themselves, actually.
That's very good. So we have the clinical practitioners, the medical practitioners, actually to do the research and study some of the basic science at the lab. So they are motivated because they observed some of the insufficiency at their own work. So I think that's part of the translational science or research.
Of course, are kind of translationals, but so in my opinion, the word of the translation research covers a very big field.
That's true.
So our translational research is to make the chemistry more happen in the medical, actually, the fields. So there are, of course, our PhD students, surgeons, can do their own surgery. But of course, they don't know much about the chemistry. That's why we need to train them. So for the first a couple of months. So after that, they can design the molecules and synthesize by themselves. So some of the surgeons now can do their own synthesis now. So this is a kind of a translational and also the multimodal and multidisciplinary. So the scientists. So now we want to the nurse this kinds of the functional the researchers, actually.
Yeah, that'd be great. So I also observed or noticed that some of the students who graduated from PhD program, they actually they go to industry for work, or they not doing research anymore. How do you see about them retaining of students?
Almost more than 90% they still do the research. So of course the medical doctors in our lab, of course they usually go back to the hospital and to keep their daily surgery. So but some of them are now had a kind of the PI positions in the hospital, and they are now starting to have this kind of collaborative work with our laboratory to develop the new types of the tools. So it is meaning maybe we can share these kinds of the motivation and passions with the medical doctors. So for the students in the pharmaceutical department, they usually go to the industry and then to keep their research in the pharmaceutical science, the company or the other companies. Maybe less than 10% they quit the research. Yes.
I see. So how do you also mentioned there are master students and a PhD student. Is the training for master students and PhD students different? Like, how do you see why?
Of course, during the master course, they need to know some kinds of the technological the means, actually. So of course, that for doing the chemistry-based medicine, the project, they need to synthesize the compounds, and they need to culture the cells, and they need to see the cells under the fluorescence microscopy, and after that they need to apply for the animals, and they need to see what happens in the in vivo situation in the animals, and after that, and they need to use a kind of how can I say, but the cancer specimen. So the fresh cancer specimen from the patients, so in this meaning the students must cover the vast field of the science.
I see.
So it will usually take time. So maybe the 2 years needed. That's why the some of the master students do these types of the training. So, but of course, the besides the training, of course, that they usually do their own research. Then after the graduate and master course, the rest with the 3 years and 4 years at the doctor course, they think about what will be needed in the future in the medical fields. And they started to design the molecules and synthesize it. And some of them are successful, that the majority of that maybe the faith. So that they go back to the molecular design and the new design, and they usually restart the synthesize. And then this kind of a cycle that they usually find out the one good way. And then apply for the cells and animals and clinical specimen for that.
I see. Do you see the number of students are increasing in the recent years, or it's decreasing?
I don't think that the number is increasing now, but maybe almost to keep
stable.
Yes, almost stable. So of course it depends on the grades actually. So maybe some years decreasing, and some years increasing, and so on. So they're almost maintain stable.
Do you think in general the Japanese young people or students from other countries, are they still interested in doing basic science research? Or they are more interested in for the AI technologies or going to the internet, those big companies?
Actually, so of course AI is the most popular the field now, and the number of the students who wants to do the AI research is increasing, but it's a kind of the fashion, I think. So for example, when I was a graduate student, so the molecular biology is the emerging field. At that moment, so almost all the students in the biological fields want to do the molecular biology. So, but now the molecular biology is so common technology, and nothing special. But of course, there are special for the CRISPR-Cas or some others, the very specific new means. So, but it's a kind of the fashion at every time of the years actually. So in this meaning, so now the AI is one of the populist, of course, the field. But maybe after 10 years. AI is the usual means for every types of the researchers. So then, I don't know. I don't foresee. I cannot foresee what will be the most popular after the 10 years. But still, they have some of the students have the passions to. So for example, of course, the AI is very interesting, even for me. But based on the AI, AI cannot tell us what types of the compounds and what types of the tools are indeed helpful for the health and the medical purposes. So of course they can suggest us, but the AI cannot synthesize.
We still need human beings to do all this kind of lab work.
So in this meaning, the chemical synthesis, this is a kind of technology. So we need to train some students very specific and they're capable of the synthesizing compounds.
I agree. So you mentioned about during your undergrad times, maybe the molecular chemistry was the hot topic. So on your scientific journey, what are the things that motivated you wanted to study chemistry?
For me?
Yeah, for yourself.
Actually, when I was a high school student, so I was so interested in studying chemistry, even for especially for the organic chemistry. So because so it's not a good way for teaching the chemistry in high school, but some of the teachers they forced the student to how can I say that, not to think logically about organic chemistry, but memorize all of the results, which is described in the text. So this kind of the history. so in this meaning that I don't want to study the chemistry in this way. So then actually, luckily, I have the very good teacher in the chemistry in high school that he forced us that you must not memorize so the content of the text. You need to think about it logically, and you can find what is the right answer for this answer. So yeah, this is very shocking for me. That was very shocking for me. But yeah, actually, we started to think about logically about organic chemistry and found that this is very simple science actually. So because of this simpleness of the organic science, so the organic chemistry, that we can design a molecule, design a functional molecules. So this is my first motivation to go into the chemistry.
I see.
So in my chemistry class in this university, I forced our students do not memorize at all. Think by yourself.
I see. I see. So maybe as our last question to wrap up this interview, to our audience, what are the things you would to share?
So in this meaning, that we need to collaborate with AIs. And actually, we have the Sugiyama sensei in the UT7. He's a very specialist in the AI. So that's why we are now collaborating with him.
Cool.
Yeah.
So we were looking forward to maybe some next outcomes from the collaboration with your team and Sugiyama sensei about using AI to find out the right probes to detect the cancer cells.
Yeah, you're right.
That would be great.
Agreed.
Yeah. Thank you so much for
Thank you very
taking
much.
your time.
Thank you.
Yeah, thank you.